Hello friends, I am Gaurang Makwana. I welcome you all to my another video tutorial of a Biotech guide. In this video lecture, we are going to discuss about an exciting and interesting topic of microbiology, that is auxanographic mutants. Okay. So what are the auxanographic mutants and what are the techniques for isolation of these auxanographic mutants? So this is our today's lecture topic. In this video lecture, we'll first brief about auxanographic mutants. What are the auxanographic mutants and their isolation technique? So these are the techniques: filtration enrichment technique, then enrichment technique, sandwich technique, replica plate, and a gradient plate. So one by one, we're going to brief about these all techniques and our main auxanographic mutants. So stay tuned and enjoy this video lecture. So let us start first. Auxotrophs. What do you mean by auxotrophs? So these are the organisms that are unable to produce an essential organic compound due to gene mutation. Okay, due to mutation or basically specific gene mutation, they are unable to produce the essential organic compound for growing. Specific condition is required, and what is the condition is there? Certain supplement. Okay, if we add certain supplement in a basic media or minimal media, so they can easily grow, but they cannot grow in a minimal media. Certain supplement is required. So these characteristics of these auxotrophs we use for figuring out the metabolic pathway. So this is the brief information about the auxotrophs. Now we'll going to further discuss about this isolation techniques for isolating auxotrophs. So let us start first with a filtration enrichment technique. Two application is there: enrichment and a filtration. So first we are doing enrichment. So we take a tube, then after we add minimal media. Okay, minimal media. Then after we added two kind of spores, mutated spores, basically auxotrophs and prototrophs. After that, we leave this tube in a shaker for the incubation. After we will proceed for the another application, filtration. So now this suspension filtrate through a filter. This one is the filter and this is the funnel. So what will happen? Basically, may be filtered through a filter such as basically this kind of filter is a cylinder glass filter, which will scan the prototrophs only. Okay, here prototrophs, because we use the minimal media. So in minimal media only prototrophs can grow, not auxotrophs. But when we add certain supplement in this kind of a minimal media, so auxotrophs can easily grow. But here which kind of media we use? Minimal media. Okay, so in result we perfectly isolated auxotrophs and prototrophs. Prototrophs is trapped on the filter, and auxotrophs that can easily pass out and collected in at the bottom of a tube is called as a filtrate. Okay, now we can grow these auxotrophs into the adenine medium. Okay, so that is specified colonies which we can collected by using a petri plate or culturing technique. Okay, so this is our filtration enrichment technique. Let's see the steps of this filtration enrichment technique. We use liquid minimal medium with inoculation of prototrophs and auxotrophs mutated spores. Then after incubation is applied and shaped for a few hours. During this time, what will happen? Prototrophs are germinate and auxotrophs will not. At the end, filtration is applied. Now this suspension will filtrate through a filter such as a cylinder glass filter, which will scan the prototrophs as a pretent and auxotrophs spores filtrate as a retent. So this is the first technique, filtration enrichment technique for isolation of the auxotrophs mutant. Second technique, enrichment technique, is basically based on a condition that affects only a prototrophic cell, not damage the autotrophs. Auxonotrophs. So basically in this technique we expose the population of this kind of a prototrophic and auxotrophic cells in a minimal media with the antimicrobial agent. Okay, so what will be resulted? So resulted a death of a growing prototropes and the survival of our auxotropes, because antimicrobial agents only affects the prototropes, not the auxotropes, because the auxotropes are, as we discussed, having a gene mutation. Okay, so they cannot be affected via antimicrobial agents. So in results, death of a growing prototropes and survival of auxotropes is resulted. Basically, this technique utilize penicillium and any another antimicrobial agent as an inhibitory agent. So this is our second enrichment technique for isolation of auxotropes. Third, we are going to discuss sandwich technique. Third is a replica, but we'll later on we'll discuss replica. First, we'll discuss sandwich, which correlate with enrichment technique. That's why we discuss first sandwich technique. As name suggests, sandwich. So here, survived mutants seated in a minimal agar, poured in a petri dish and covered with a sterile medium. As you can see, this is the petri dish. Okay. At the bottom, survived mutants seated in the minimal agar. Then after we'll covered with a sterile medium. Okay. After plate is incubated for 12 days. After 12 days, we observe the developed colonies and we mark it down at the base or the top. Okay, we mark it down the developed colonies. After that, we applied a layer of a supplemented agar on this surface. Okay, so this is the supplemented agar we applied, and after same incubation is applied, and after incubation, colonies of auxotropes were unable to grow on the medium, and this kind of a visual identification which we observed, which is basically used for the auxotropes identification. Okay, so basically the technique is simple. None of any kind of instrumentation or laborious work is there. Only the plate and a medium is required. Okay, so this is the sandwich technique used for isolating the auxotropes organism. Now next, our most important technique, that is the replica plate technique, developed via Joshua and Esther Ledgerbury, scientists, 1952. And this technique is basically used for the screening technique. Basically, is the screening technique used for the recombinant DNA, which copies the pattern of colonies from a growing culture. Basically, okay. So this technique is used for a kind of a auxonographic isolates mutants. So technique used for the detect auxotrophic mutants and a wild type of a strain on the basis of ability to grow in the absence of amino acid. Okay, so here one application is there, absence and presence of amino acid, medium having one medium having amino acid and another is a lack of amino acid. So based on that, we identify or detect the auxotroph mutants and a wild type of a strain. Okay, so this is the basic principle. Also, this technique is used for demonstrate the presence of antibiotic resistance in a culture. Okay, and also this technique having application of inoculating needles as a velvet cloth. Okay, one wooden block is there, is covered with a velvet, and that block is act as a inoculating needles for the mutants, which can be replicated and isolated. And also we have discussed that this is the screening technique for the recombinant DNA, copies the patterns of a colonies from a growing culture. Okay, so this is a brief intro about the replica plate. We'll further going to brief in next what is replica plate technique. Now we'll see the steps of a replica plate technique. So some steps are needed for this replica plate techniques. First step is to what? Take a master plate and copy them into another plate containing a different medium. Then after, second step is what? With the help of a sterile velvet on a wooden block, take the colonies from a master plate and imprint of all colonies into another fresh media. Okay, and that's fresh media is we take two qualities. First is a lack of ampicillin and another is a contain ampicillin medium. Then third step is what? Incubation. Okay, we incubate these two plates and we'll see the result. What will happen? Let us revise the technique with a diagram. We first take a master plate and copy them into another plate, which is this. Okay. After that, we take a wooden block with a sterile velvet. This is the wooden block and this is the sterile velvet, and we imprint them into another two kinds of a media. One is having a lake of ampicillin and another having a ampicillin. Okay. Then after we incubate these two plates and we'll see the result. So result is, as you can see, a plate having a lake of ampicillin, having a ampicillin sensitive colonies like this. This is the lake of ampicillin plate, and colonies generate which having a characteristic of a ampicillin sensitive, which is sensitive to ampicillin antibiotic basically. Then another one in second plate which having a complete amount of ampicillin produce a ampicillin resistant colonies. This kind of a colonies is resistant to a ampicillin antibiotic. So this is the simplest and screening technique basically for the isolating the auxonotropes and also inoculating the mutants from the master plate. So this is the fourth and important technique for the exam purpose, replica plate technique. Then we are going to discuss next fifth gradient plate technique. As name suggests, gradient is generated like you can see in this diagram. Bottom layer contain nutrient agar and top layer contain a nutrient agar with an antibiotic. Okay, we labeled with a positive which having a antibiotic, and at the bottom which doesn't having antibiotic. So this kind of a gradient is generated and we observe the result. We are inoculating the organism. What kind of effect are there? So this technique, basic principle, having ability to organism to produce mutants that are resistant to antibiotic is grow them on a gradient plate of a particular antibiotic. So this is the basically technique for the finding out the resistant organism. Generally we call them antibiotic antimicrobial resistant technique. Also technique consists of two edges like a layers, as we discussed. Top layer having antibiotic and a bottom layer doesn't have antibiotic. So this kind of technique is there. We'll further going to brief in next. Now we'll see the process. 5-step process are there. First is to autoclave the two nutrient agar tubes. We'll set it for the 55 degrees centigrade. Then we taken out first tube which will pour in the plate. Then we generate a gradient. We are using a glass rod. We set the glass rod at the bottom of a petal plate and solidify them for the 50 minutes. Then we take another tube, mix it with streptomycin solution. Then pour it with the same procedure on the bottom layer. Then fourth step, marking. We invert the plate and mark with the LSC. LSC stands for a low streptomycin concentration and HSC stands for a high streptomycin concentration. Then fifth step, inoculation. So we taken out 0.1 ml of a test E. coli solution on the plate and incubate for 37 degrees centigrade 24 hours, and we'll observe the result after incubation. After incubation, we'll see the result. What will happen? So basically, let us revise again. Our top layer is defining as HSC, high streptomycin concentration, and the bottom layer is defining as a LSC, low streptomycin concentration. After incubation, what we observed, colonies that are grown in a HSC marketplace here, that is defined as a streptomycin resistant colonies because they are producing mutants against streptomycin. Okay, so those colonies are defining as a streptomycin resistant colonies. And another colonies produced on the LSC area, LSC marketplace area is defining as a streptomycin sensitive colonies. Okay, so this is the gradient plate technique. And here we completed our video. I hope you enjoyed very well and got it very well. Thanks for watching. And if you want to get more and more interesting videos like this, Join with us and be a perfect biotechnologist. Thank you so much, friends.